you remember a big sphere, you can close a knot a little bit on the close to the North Pole or a little bit closer to the South Pole. So we have a young Grainge from Australia, and so I hope that closing at the South Pole will come to the same result like closing to the North Pole. So let us hear.
Yeah, thank you for the introduction, and I will come back to the kind of reactions that Sean was talking about at the end. So don't forget about Pepe and this wrapping structure. I will come back to it at the end of the talk. But what I'll spend most of the talk on is reactions which are slightly different. So Xer can do a different type of topological reaction, and it's cartooned here. This is another sort of monster Frankenstein-like structure. It's not actually one crystal structure, it's bits of different crystal structures stuck together. And here you've got a site-specific recombination synapse with 2 pieces of DNA. And over here you've got this motor protein FTSK. And I'll go on and try to explain to you how these things interact and how they lead to a different topological outcome for recombination. So just a bit of allergy advice that this talk will contain gels. And if you're already feeling a bit queasy after Sean's talk, then you might want to run screaming for the door or have your adrenaline shot ready. So a bit of basic biology background just to reiterate what Sean's already said. Bacteria tend to have a circular chromosome. Of course, like any population, there are weirdos out there, and some of them have linear chromosomes, but most of them have a circular chromosome. And most of them replicate starting from one point and replicate round bidirectionally, finish off somewhere opposite the origin, and what you get out is 2 circles of DNA. And the idea is that then you divide the cell down the middle, and each daughter cell gets 1 full chromosome's worth of DNA. However, replication, like true love, doesn't always run smoothly, and so it runs into problems. And these problems can be corrected by homologous recombination. And about 17% of the time, these homologous recombination events lead to a crossover. And if you have a crossover, what you get out of the end of recombination isn't 2 monomers, you actually get a chromosome dimer. So 4.6 megabases of DNA in each of these things actually join together into 1 massive dimer of chromosomes. If you've got a chromosome dimer and you try doing cell division, you can't equally put the same amount of genetic material into the 2 daughter cells. You either end up with a dimer in one cell and nothing in the other, or you slice the chromosome randomly in half and each daughter inherits a broken, rapidly degrading chromosome. And either of those leads to cell death. So if you have a chromosome dimer and you don't resolve it, then you die. So bacteria have this Xer recombination system, which doesn't just work on plasmids. It actually probably evolved to resolve the chromosome dimers, which takes a dimeric chromosome, does a recombination reaction between these specific sites, and converts the dimers back into monomers. Once you've got that, you can lay down the septum, divide the cells, and you're back living. The dimer resolution reaction has slightly different requirements from the resolution reaction on the plasmids that Sean was talking about. So you require a small site, diff, which is only 28 base pairs. You don't require any other accessory DNA sequences. So there's no binding sites for Pepe or RJAR or any other proteins that are known. And you can take that sequence and put it into a completely different context and it still works. So we're pretty sure that there are no accessory wrapping proteins. You require the 2 recombinase proteins, XCRC and XCRD, which recognize this site and do the cutting and exchanging of the strands. And you also require this protein FTSK. So just to go over what Sean's already told you, in the recombination reaction, you have these 2 recombination sites, which I will always show with these little red triangles, and they come together. And each site is bound by a monomer of XCRC and one of XCRD, and protein-protein interactions bring the 2 sites together. You get cutting and exchange going through a holiday junction, and you end up with a different configuration like that. And this shows the crystal structure of the Cree recombinase in the middle of doing this reaction. So that's the cartoon, that's what the actual protein looks like. So it's been known for a long time, since 2002, that if you do this reaction in vitro using a plasmid with 2 diff sites, shown here, and you add XCRC and D, and you add this protein FTSK, and it requires ATP for activity, you add all these things together, what you get out is exclusively unlinked products. So in the reaction Sean was showing you, they are enriched for 4-noded catenanes. In these reactions, you don't get any catenation at all, but only simple products. And the question is, how does FTSK influence the reaction to give you only unlinked products? And the answer is I don't really know, but I'll give you some kind of hand-waving explanation to try to explain it. So the FTSK protein. FTSK was identified in 1995 in a screen for proteins involved in cell division. So the screen was to make temperature-sensitive mutants which at a low temperature were working and the cells would grow happily. And you switch to a high temperature, the protein becomes inactive, and the cells can no longer divide, and you get these massive long sausages of filaments of cells, and eventually they burst and die. And so this protein FTSK stands for filamentous temperature-sensitive mutant K. So FTSK somehow is involved in cell division. And it turns out that FTSK is a rather large protein, and it does multiple different things in the cell. So you can divide it up into 3 basic segments, a red, green, and a blue segment. The red segment spans the membrane and interacts with various other proteins in cell division. So it interacts with one of the first proteins, which identifies the middle of the cell, the point where the bacteria wants to divide. It then gets localized to that and then acts as a scaffold to bring in lots of other cell division proteins. And these are cartoons showing where it interacts with all of these different proteins. So it interacts with all of these FTS proteins, FTSZ, Q, L, I, and so on. So most of the interactions are in this red region and in this green cytoplasmic region. And these are the regions that are vital for the cell to grow. But FTSK also has this blue blob on the end. This is a very low resolution structure. So this blue blob is sitting out in the cytoplasm, and it's this blue blob which is actually involved in the XCR recombination mechanism. So if we look at a slightly higher resolution structure of this, what we're interested in for the rest of the talk is what was the blue blob on the previous slide, and now you can further subdivide that into domains. So we've now got domains which we call alpha, beta, and gamma. And we have crystal structures of each of these domains. So shown down below is a crystal structure of alpha beta. And alpha beta comes together to form this nice 6-fold symmetric hexameric ring. It's a ring with a big hole down the middle. And double-stranded DNA fits down the middle of that hole. You can also see maybe these little yellow blobs at the interface between subunits, and those are the nucleotide. So the protein sits on double-stranded DNA, it binds ATP, hydrolyzes ATP, and uses the energy from that hydrolysis to move along the DNA. And it actually moves at the fantastic speed of 17,000 base pairs per second. So if you stretched out 17,000 base pairs of DNA as linear, it would be longer than the E. coli cell, much longer. So it can move a distance equivalent to larger than the whole cell that it's in. If you scale it up to be the size of a sort of family saloon car, it would be going at about 3 times the speed of sound. So it's pretty nippy protein. The actual alpha and beta domains sit on top of each other and form 2 rings, 2 separate rings, and the beta domain is the main motor part of this. And then sitting on the end is this small blob, which we call gamma. And it's known that gamma also has a couple of roles. It's involved in orienting the motor in the first step. So the motor knows where it's going as well. It seems to be a clever motor. It doesn't just buzz around the DNA any old where. It always loads so that it goes towards this recombination site diff. Okay? And it uses the domain gamma to do that. And the small gamma domain, it turns out, also can interact with the recombinase XCRD and seems to stimulate recombination. So if you delete that motor domain, then the cells live, but they don't look entirely normal. So you get a mixed population of normal-ish looking cells. So these are wild type. Here you've deleted just this motor domain over here. And some of the cells look like long filaments, and some of them look like sausages, joined together, links of sausages. These cells probably can't resolve chromosome dimers, and these links of sausages are delayed in cell division because they've probably got bits of chromosome trapped where they're trying to divide, suggesting that FTSK normally is sitting at the septum, pumping DNA away so that you can then divide your cell without trapping any DNA. So again, just to go over that schematically, you start off with a newly born E. coli cell with a blue chromosome. You grow, you replicate, segregate to the opposite poles. If you remember Nancy's talk from Monday, she showed images of this occurring. You then start to lay down a septum, and at this point FTSK gets recruited, becomes active as a translocase, and pumps any DNA away that happens to be trapped there. And then you can divide the cell. So FTSK is doing multiple roles in the cell. It really is a fantastic protein. So it acts to recruit other factors for cell division. So it's a key divisome component. It pumps DNA that's being trapped at the septum. It knows where it's going, so it always pumps towards the diff site. And by doing this, it can actually bring two diff sites together. And then it seems to stimulate the recombination between these diff sites so that you can then segregate your chromosomes into opposite poles. Another thing it can do is help the unlinking of the chromosomes. So it can do this both directly and indirectly. So it does it indirectly by probably interacting with topo4, and it can either just stimulate topo4 activity, or it just smushes all the crossings into a smaller area so that topo4 is more effective. It can do it directly by stimulating multiple rounds of XCR recombination, and the change in linking number has been shown to be able to uncatenate chromosomes as well. And this is the proof of how you can go through this decatenation reaction, which I'll go through. No, I won't go through, it's just joking. This is a biological talk. That was just to wake up the mathematicians. Back to the biology. So FTSK, you do the reaction in vitro, you take a plasmid substrate, and you've got these two recombination sites. You add FTSK, ATP, and somehow you only get unlinked products. So how is this DNA translocase, a motor protein moving along DNA, giving you this reaction? There were various models put forward that perhaps the ATPase activity of this motor is involved in somehow remodeling the complex, or that the positive supercoils generated by its movement might help stimulate the recombination reaction. This turns out not to be true. So this is just an experiment to show that all you need to stimulate the recombination reaction is that small domain gamma sitting on the very end of FTSK. So this part here, so the final 70 amino acids. So what you do is you grow up a reporter plasmid with two diff sites in a cell, and if XCR recombination is active, then it resolves these into two smaller plasmids. And what you do is that you make the strain deleted for that C-terminus of FTSK. And then in that background, you then can express at a certain time just that gamma domain. And over time, as you express that gamma domain, you switch from the reporter plasmid here into the recombinant products. And so it shows in vivo that if you have just that little gamma domain, suddenly you get recombination, whereas in the FTSK minus background, you would otherwise have none. So all you need for stimulating Xer recombination is gamma. For various reasons, partly because we could do it and it was fun, partly because it makes it more stoichiometric, the amount of gamma interacting with the Xer recombinases, we made these fusion proteins. So you genetically just clone the protein with the gamma domain stuck on the end, express it as a fusion, and you can just purify those proteins and use them in vitro. And it turns out that if you express these proteins, this is again in vivo, they're incredibly active at recombination. So here we've got the same thing. You've got a strain which doesn't have the FTSK motor domain. You start expressing these fusion proteins and look after a certain amount of time, and 100% of that plasmid has been converted into the recombination product. So these are incredibly hyperactive, self-activating mutant fusion proteins. And I'll refer to these as either fusion proteins or the hyperactive fusion proteins. So we can then ask, if we've got these hyperactive mutants, are they working normally on the chromosome? And we can do this by taking various different strains. So we take a strain where we've knocked out XERC and then try expressing this XERC fusion protein in that strain. Now normally, if you've got an XER mutant or an FTSK mutant, about 17% of those cells, growing as they are, go through this dimer pathway, and without being able to resolve that dimer, they die. And so you get a fitness loss from being XER minus. And you can use that as a way of seeing whether your protein is complementing that fitness loss. So in an XERC mutant, you can express wild-type XERC, and you recover that fitness loss. You can express the C-gamma fusion protein, and again, you recover that fitness loss. So it's working in recombination on the chromosome, just as XERC would. You can do the same with XERD, and you recover the fitness just as you would with the wild-type protein. If you now do it in an FTSK delta-gamma background, so these proteins I've shown you before don't need FTSK anymore to recombine. So they can recombine without FTSK there, but in the absence of FTSK, you don't recover that fitness loss. So the proteins are capable of doing recombination without FTSK, but it doesn't get you out of this chromosome dimer resolution mess. So either the two diff sites in that context without FTSK aren't coming together efficiently, or they're coming together and recombining to give you something horribly knotted, which the cell then can't deal with. Or it's a combination of the two things. So FTSK could be working to pump the DNA so that you align these sites so that you get recombination efficiently, and the recombination products are unlinked. So here's just a little aside to wake you up. This is E. coli cells growing. So this is the experiment shown here, where you're complementing an XERD mutant with this fusion protein. Ooh. And this is time-lapse of these cells growing, and you see them elongating and dividing, and the vast majority of them look like normal happy E. coli cells. Seems to be running very slowly, I apologize for that. But occasionally, if you keep your eye on this guy here, you'll find that one of them grows into a filament. Now, the really odd thing about this background is that this filament then actually starts dividing. So it's divided at that end. You let it grow a bit more, it's going to divide there. Still a big long filament along there. Let it grow a bit more, dividing again. Divided there, and then finally it divides in the middle, and it's no longer a filament. So it's gone back to being a perfectly normal cell. So it seems that possibly this hyperactive Xer recombination activity might be causing possibly a topological problem of the DNA. That means that it takes time for the cell to untangle all that DNA before it can then segregate the chromosomes and divide. And eventually, when you let these things grow, they go out of focus because cells start living on top of each other. So FTSK seems to be necessary for driving the reaction to give you free, unlinked products. So Sean already introduced the idea that with other recombinases, what you can do is just bring sites together randomly, and what you should generate is a whole slew of different catenated products. And the products depend on exactly how many of these crossings you trap when you bring the two sites together. So, what happens now if we try our hyperactive mutants on these substrates? So they don't require FTSK anymore. And the answer is, shown in this gel, you start off with, this is now a nicked piece of DNA to remove all the supercoiling, and that then things run basically depending on how many crossings are trapped. So these are either catenation or knotting crossings. And what you get is, over time, you see the appearance of these bands, and these bands correspond to the whole load of different catenated products. So hyperactive Xer fusion proteins make catenanes and knots. So we've gone from a nicely controlled reaction under control of the FTSK to loads of catenanes. So it must be the translocase activity of this protein that's somehow leading to unlinked products. And we can show that again. So here we've got, unfortunately, 50C means FTSK. It's just lab jargon. I apologize for that. Here we've got a plasmid which is being recombined by Xer C and D in the presence of FTSK to give you these two free products. If we do the reaction with these hyperactive mutants, we get recombination, so you can cut the reaction and see that there is recombinant product, but they're all catenated and running about the same size as the supercoiled. We can then take this reaction and say, what happens now if we add that motor protein back? And you convert from catenated products into free products again. So the motor protein is somehow bringing the two sites together so that there's no crossings trapped between them. We then did the experiment where you add back not just, well, you add back just the motor protein without that little gamma domain added on. So these fusion proteins are self-activating, so they shouldn't need the gamma domain from FTSK to react. But if you have the intact motor, you get lots of free products. If you have just the motor without the gamma domain, you get a bit of free product, but a lot of catenated product. So it seems that it's not just translocation activity, it's translocation activity of a motor which has gamma attached to it that's important. And that's a bit hard to imagine why. And you can do this with all the other various combinations, and it gives you the same results. So, just to summarize what I've told you, it is rather complicated. FTSK with Xer-CD produces free products. All that you need for the stimulation of recombination is that gamma domain. However, the FTSK gamma domain alone with Xer-CD gives you complex products. And you only convert back to simple products when you've got the full alpha beta gamma motor. So why is that? So this is the sort of hand-waving, take it or leave it, believe me or not, explanation. So if we've got a plasmid here with 2 diff sites, and it's supercoiled, it might sit like this or be branched. We then add XCRC and D, and then add FTSK. And it could be that FTSK sits on one strand of DNA, and then with its gamma domain sort of reaches across and finds a recombinase to interact with. And if it reaches across and finds one over here, and then activates its translocase, it basically reels in the other site. It then stimulates recombination between these things, and you get unlinked products. So that's all well and good, great model. However, the problem comes at this stage. Why do you only get simple grabbing? So you could imagine that at this stage you could grab something over here and trap lots of supercoils, and then when you recombine you again get complex products. But it seems not to do that. And the hand-wavy explanation could be that either because it's traveling on DNA, it's increasing the slithering of DNA, so the 2 segments slither past each other more efficiently, and it's then sampling these slithering other segments more quickly than it's sampling in 3-dimensional space. Or it could be also related to the speed at which it moves on DNA, which is incredibly fast. So these plasmids are roughly 5,000 base pairs. It's moving at 17,000 base pairs a second. It will sample the whole plasmid in under a second. So it could be sampling adjacent slithering pieces of DNA very quickly and possibly faster than 3-dimensional movement. That's the hand-wavy bit. I don't really know if that's true. But it's a nice model. So does FTSK translocate so rapidly that it captures these things without capturing any topology while it's doing it? Or it could be that FTSK is in some way set up that it will only activate those synapses which are simple. So saying that perhaps it can see external topology and it knows what it's doing. And I think that's really attributing too much intelligence to the protein to do that. Or it could be that FTSK somehow disassembles complex synapses. And again, it's hard to imagine how it could know which ones to disassemble and which ones to activate. Another piece of evidence that we gathered to sort of say that FTSK doesn't really know the external topology is it can capture linear diff sites and recombine them. So you can force it by having lots of these little diff sites to recombine inappropriate things, suggesting that it can't really sense external topology. And I won't go through the gels to show that. So to conclude that part of the talk, that the gamma XCRD interaction is all you need for stimulating recombination, and that somehow translocation of this entire motor goes towards giving you free circle products. The next thing was looking at how the recombination reaction actually goes in the presence of this gamma protein. So it's been shown that for the reactions that Sean was talking about on the plasmids, that there's a hierarchy of which protein likes to act first. So in the Pepe Psi reaction that Sean was talking about, XCRC cuts first and then makes a holiday junction, and then XCRD resolves that holiday junction. And it turns out that in the FTSK stimulated reaction, it's the opposite way round. The XCRD goes first, and then XCRC resolves that holiday junction. And we can show this directly by adding peptides, which Anka Siegel made, which happen to trap that holiday junction intermediate. And so you can do reactions with these hyperactive mutants and trap the holiday junction, get incredibly complicated gels, which I won't bore you with, but the outcome is that in the presence of gamma, XCRD makes those holiday junctions at diff. So We don't exactly know how gamma is stimulating XCRD to make those holiday junctions, but it seems to. So now, what happens when we go back to a reaction which is more like what Sean was talking about? So we've got the Sean setup where we've got the psi sites. You get these 3 crossings trapped by interwrapping of the PEP-A protein, and it gives you a 4-noded catenane. So what happens then if we add our hyperactive mutants onto this site? The outcome is that, again, these hyperactive mutants will recombine that to give you a whole load of different catenated products. But if you add PEP-A back to that reaction to give you this interwrapping, then it gives you a 4-noded catenane again, or at least massively enrichment for the 4-noded catenane. So these hyperactive mutants are responding to the external topology of the DNA just like the normal proteins would. But I've told you that in the normal reaction at psi, XCRC likes to go first to break the holiday junction, whereas with these hyperactive mutants, XCRD likes to go first. So the question is, in this situation, what wins? The protein being hyperactive or external topology? And the answer is that topology wins. So it's a victory for topology. So that if you have these hyperactive mutants, then XCRD cleaves to give you that holiday junction product, and if you have a mutant D which can't cleave, you get nothing. If you have the mutant D which can't cleave, and you add PEP-A to it, then you get lots and lots of holiday junction product. And so it seems that having these 3 interwrapping nodes outside it brings the sites together in such a way that XCRC will like to recombine first. And it overrides the protein's desire for XCRD to recombine first. So that synapse must be a pretty tight entity, bringing it together so that the order of strand exchange is already determined by that synapse. And that's just a summary of what I've said, that these hyperactive mutants are active at the psi sites to give you complex products. You can convert this complex slew of products into a 4-noded catenane by adding PEP-A. And that the external topology seems to control both the synapsis and the recombination mechanism of these proteins. So just to finish with acknowledgements, thanks to David Sherratt, whose lab most of this work was done in, and collaborators in Oxford, Christian and Mark Howarth. And the crystal structures I showed you were by this guy, Jan Lerver, who's here in Cambridge. And thanks to the funding bodies. And now just a plug for the Southern Hemisphere. This is Newcastle, beautiful views, empty beaches, rainbows every day. And we do have lectureship positions and PhD positions available in Newcastle if you want to come and enjoy the weather. Thank you for your attention. Yes.
So what happens in vivo when you don't have the diff site?
If you don't have a diff site, then 17% of those cells die because they can't resolve their chromosome dimers. The rest of the cells look fairly normal.
But if you look in more detail, what happens with your proteins? With mutations?
They don't seem to recognize any other any other sites, so they're not doing inappropriate things in the absence of their proper site. And you can really overexpress them, and they don't do inappropriate recombination reactions.
Do they still translocase, though, if they don't have the diff to run to?
If FTSK, yes, FTSK does still translocate. So it doesn't require the diff site to translocate. It reads sort of polarized sequences on the DNA so that it knows which direction to go in anyway. And it will pump the DNA and help DNA segregation, even though there's not a diff site there.
Then what happens?
We don't actually know what happens, but we know that this is an incredibly fast and incredibly powerful protein that's capable of kicking off any other protein we've tried on DNA, and it can break the biotin-streptavidin interaction. So we think that it's going along the chromosome and kicking off anything that it meets, and then when it gets to the diff site, it interacts with XCR-C and D and actually stops. So if the diff site wasn't there, it might sort of go on a bit further, and you might think that you might cause problems with it sort of kicking off proteins that it shouldn't or going too far, but there's no gross phenotypic change that we can see.
If the diff site is in the TER region and this protein is going through that and it's polar, when it goes through the TER region, it's now antipolar. Isn't it going to recognize it's driving on the wrong lane of the motorway and it's going to stop?
It seems that it only sees the DNA at the loading step. So it seems that it can then go through these polarized sequences in the wrong direction and it doesn't see them anymore. So it knows where to go at loading, and then it's blind. It might run into a hexamer going from the other direction, and the two things stop each other. Or it might just go and cause problems. I don't know.
Okay. So, yes, sorry.
One of the slides, I wasn't sure if you had sites on linear bits of DNA, and I think the question was if FTSK could identify those linear ones that weren't connected to the main plasmid. This was to try to understand whether it's reading the global topology or what is it doing. I was just wondering, I didn't understand what the conclusion was of that.
So if you have a high enough concentration of these small linear bits of DNA, FTSK can force them to recombine. So the conclusion is that FTSK doesn't know the external global topology, and therefore how can it know which ones to, it can't be selective in which synapses it's going to activate. It must be just bringing together only simple synapses in the first place, was the conclusion.
In your very last experiment, you add Pepe and it reverts to going C first and then B next. Why do you think that is? Do you think the global geometry has something to do?
Yeah, I think it's that the DNA is wrapped in such a way that the two sites come together in the geometry which only allows XCR-C to go first. So it must be a really tight, defined geometry, and it can't switch to the other geometry where XCR-D would want to go first, as the proteins would want to do anyway. It must be that the external topology is altering the geometry of the two sites very locally. And it would be interesting to then sort of put in space of DNA between that and the recombining sites and see whether you now relax things and then XCR-D goes first again. But I would think that it means that the psi site is really interwrapped so tightly that the two sites are really juxtaposed. And that would fit with Sean's knotting experiments as well.
Can the relative position of the sites be swapped with respect to the accessory sequence for Pepe? And do you think that would make a difference?
It's a question for Sean, really. I was speaking to Sean the other day, and he said that they've added in bases between the sites, or you can flip the sites, and you do alter the activity. But I think if you add in much more DNA between the two sites, then in the normal side reaction you start losing recombination products pretty quickly. You also see the phasing. So if you add in 5 bases, then you get very little. If you add in 10 bases, then you're back into the correct phasing.
Well, more than adding DNA, I'm thinking just changing the order of the accessory sequence. Instead of having C, the accessory sequence, having D, C, accessory sequence.
Yeah.
So the accessory sequences make the recombinase that's closest to them go first. So you can do just that experiment.
And then make D go first.
You can make D go first and by flipping the D and the C around.
So I will have short announcement after we thank Jan again.